Hi everyone, welcome to the BGE Navigator tutorial series. In this video, we are covering the browser tab. This is where everything comes together. After picking your cell types and selecting your tracks, you are now going to visualize them in the Washu Epigenome browser. This is the core visualization engine of the portal. Let's dive in. When you click on the browser tab, or when you click Next from the Asset Selection tab, you will see the virtual epigenome browser loaded with your selected tracks. At the very top of the browser panel, there is a small header with the virtual epigenome browser logo and title. Next to it, you will see a full screen button. We'll talk about that in a minute. If you switch to another tab, then come back. The browser remembers where you were. It preserves your last view region, so you won't lose your place. Now here is something really important to understand. The browser always uses human or HG38, the human genome assembly, as the primary references. All tracks are displayed in HG38 coordinates. But wait, what about mouse marmoset data? Here is a clever part. The portal automatically adds a genome align track for any non-human species in your selection. So if you select some mouse taxic tracks or histone modification tracks in this example, you will see both sets aligning to the same genomic locus through the genome alignment. This means you can directly compare epigenomic signals across species at syntenic regions. Besides your selected data tracks, the browser comes preloaded with a set of default annotation tracks, including ruler, RefSec gene annotations, and AAV enhancers. They are always presented regardless of which data track you've selected. They provide essential context for interpreting your data. Let's show you how to navigate around the genomes. Click and drag left or right to pan along the chromosome. This is intuitive, just grab and slide. Use this tool to zoom in to a region or zoom in from here to here. You can also jump to a different region using the tool here. You can also use the same tool to search for a gene. You can zoom out a little bit to see the whole scope of the gene. All of these navigation tools are standard virtual epigenome browser features. So if you've used the browser before, you will feel right at home. The browser panel also includes a full screen feature that I really recommend using, especially when you have too many tracks loaded. Just click on the full screen button or press F to enter and exit full screen. It will give you the maximum space for your track. This is great for presentation where you really need to study the data closely. Inside the browser, each track is rendered individually. You can change the display option by right-click each track name and make changes. The WashU Epigenome browser has extensive documentation on these features. There is a helpful link right below the browser view and says browser documentation. It will link to a GitHub page where you can learn more about every feature in detail. That's a browser tab. In the next video, we will cover the single-cell analysis tab for single-cell analysis. Thank you for watching.